When you get a paper cut, the top layer of skin can quickly heal back. However, when a deeper cut is made, the skin heals but leaves a scar. Scar formation is the outcome of most wound repair in humans. In contrast, salamanders can heal without scars and regenerate. What is regeneration? Regeneration is the process of restoring lost tissue or organs. One species of salamander, the axolotl, has been used in scientific studies for decades because it can perfectly regenerate its limbs, tail, brain, spinal cord, and sections of its heart throughout its lifespan. How do axolotls pull off such an incredible feat? When a limb is lost, the axolotl bleeds very little, and the wound seals off within a day. Cells migrate to the amputation site and form a special mass called the blastema that will give rise to a new limb. Mammals are incapable of forming a blastema after the loss of a limb, which would be a key step towards successful regeneration. Since salamanders and humans evolved from a common ancestor, it is possible that the ability to regenerate is present, but locked away in our genetic code. There are three main phases of regeneration: wound healing, blastema formation, and patterning and scaling. In the wound healing phase, cells at the wound edge release damage-associated molecular patterns, DAMPs, which are immediate signals that quickly reach nearby cells. These chemical signals activate skin cells called keratinocytes to crawl over the open wound. Within 10 to 24 hours, the wound edge is sealed. DAMPs also signal to immune cells called neutrophils and activate them to help fight infections. They are the first responders to injury and are quickly recruited to clear out cellular debris and pathogens. Another type of activated immune cell is the macrophage. In mammals, macrophages often drive scarring after the wound healing phase. But they also have regeneration-promoting abilities. In the axolotl, macrophages are essential to regeneration. During the early stages of limb regeneration, one of the macrophage's roles is to release digesting proteins called matrix metalloproteases, or MMPs. MMPs chew up the extracellular matrix, ECM, which provides structural support to the surrounding cells. Thus, cells near the wound are liberated from the rigid tissue scaffold due to ECM remodeling. Macrophages coordinate and direct other cells to promote scar-free healing and form the blastema in the first week after injury. Throughout the formation and growth of the blastema, the characteristic process of cellular dedifferentiation occurs, whereby differentiated cells, such as fibroblasts, revert to an unspecialized state. Like embryonic cells, dedifferentiated cells can give rise to different tissues like cartilage, tendons, and joints. Over weeks, the blastema continues to grow as the patterning and scaling phase begins. Cells in the blastema turn into tissues needed to form a new, perfect, yet tiny limb. In 3 months, the new limb grows to the right size for the axolotl. It remains a mystery as to how the limb knows when to stop growing once it has reached the correct size. There remain many questions to be answered by scientists, and the most pertinent of all to human health: are there signals that can provoke regeneration in humans? As more is revealed about the axolotl's regeneration process, the knowledge may one day allow humans to recover or regenerate without scarring through chemical and cellular therapies that activate tissue regeneration.